Hello, thank you for joining. My name is Rika Gatesi. I am a product development scientist at Precision Nanosystems. Today, I will present to you a novel technology for the robust, low-volume production of lipid nanoparticles for nucleic acid drug discovery and screening. Our company's mission is to accelerate the creation of transformative medicine that significantly impacts human well-being. In treating diseases at its fundamental level, genomic medicines are the future. The genomic medicines approaches include silencing, expression, and editing of genes. Thus, this allows for the targeting of any gene, any way. Importantly, genomic medicines are designed, not discovered. They are validated and manufacturable, thus ready for mainstream use. The successful development of genomic medicines needs 4 pillars. First one is to define a specific gene target, whether this is to silence, express, or edit, and then we choose an active pharmaceutical ingredient, or API, which is a nucleic acid as a payload here. This is followed by choosing a delivery carrier or a vector, such as an LNP, to protect and transport the payload. And finally, we need a scalable manufacturing solution. Here, our proprietary lipid nanoparticle platform, GenVoy-ILM, solves the nucleic acid delivery challenges, and our proprietary microfluidic-based manufacturing system, the NanoAssemblr platform, solves the drug product manufacturing and scale-up challenges. Importantly, our team at PNI has the expertise and experience to solve the knowledge gap. There are various strategies for the introduction of nucleic acids into the cells in order to modify their activity for research and clinical purposes. To achieve this, nucleic acids need to penetrate the cell membrane and enter the cytoplasmic space. Some of the commonly used approaches involve mechanical disruption of the cell membrane, such as electroporation. Others include viral delivery methods or chemical means, such as liposuction. These conventional approaches have both pros and cons that may limit their suitability as a gene delivery method. For instance, unprotected genetic material is rapidly degraded, while compromising the cell membrane leads to poor viability. Viral and cationic lipid-based delivery modes protect the payload, but have limitations in terms of scale-up and in vivo delivery. Overall, these methods are not ideal, and there is a need for improved gene delivery solutions. You can see some of the key disadvantages listed in orange. Several physical and chemical properties of LNPs lend themselves to be effective vehicles for nucleic acid delivery. The dense packaging of payload in the core of LNPs protects its integrity as it enters the cell. The lipid outer shell allows for cellular uptake via endogenous mechanisms and hence has very low toxicity for sensitive cell types. They are also very easy to use and translatable from bench to bedside. Thus, you can implement LNP technology from in vitro to in vivo to in human use in a commercial vehicle application. Precision Nano's current next-gen microfluidic mixing technology is the advancement of its previous staggered herringbone geometry and is the most advanced in the market. This overcomes the shortcomings of traditional techniques, such as T-tubes in lipid nanoparticle formulations. It is an innovative platform that offers an ultra-low volume LNP formulation process and at the same time gives rise to scalability and high throughput using the same next-gen microfluidic technology. It is precisely designed for non-turbulent particle formation to ensure the most reproducible results for a wide range of nanoparticle types. The next-gen microfluidic geometry that I just described is at the core of the NanoAssemblr instrument lineup. These instruments are all equipped with the same mixer, but give rise to various scales from discovery to commercialization. This allows for a consistent process throughout your drug development program, allowing for seamless manufacturing scale-up. Today, I will talk about the NanoAssemblr Spark instrument, which is positioned for drug discovery and screening at the starting phase of preclinical research. The NanoAssemblr Spark is ideal for discovery and early preclinical development of mRNA lipid nanoparticles by enabling rapid, reproducible, low-volume formulations to conserve limited or expensive ingredients. The Spark is suitable for the controlled manufacturing of LNPs at volumes from 50 microliters to 250 microliters, encapsulating only a few microgram of RNA with minimal waste. You can treat your cells directly as it comes out from the instrument, thus allowing for easy integration into cell culture workflows. Moving beyond the traditional methods, such as T-tube or T-junction geometries, a researcher can use the Spark instrument as a more convenient and more reproducible formulation of lipid nanoparticles. Applications for the Spark includes nucleic acid delivery and screening, nanoparticle design and screening, drug discovery, and target validation. So how does the Spark remove research barriers? With the success of mRNA COVID vaccines, many organizations are working with lipid libraries in addition to having a large library of payloads. These two parameters together, as you can imagine, can generate hundreds of formulations, which means large experiments are needed for finding optimal characteristics, such as biological activity. What this means for researchers is a large amount of time and financial investment, particularly when dealing with expensive materials like that of mRNA. As the Spark takes few seconds to formulate and the resulting LNPs can be applied to cell culture without any downstream processing, it offers a very efficient workflow along with a significant reduction of hands-on time. On this end, formulating lipid nanoparticles on the Spark is as simple as pipetting. Here you would fill in the cartridge with the input solutions, put on the cap, insert the cartridge into the instrument, and hit the start button. The mixing process takes less than 10 seconds, at which point the nanoparticles are in the output well and are recovered and optionally diluted. The Spark cartridges with the next-gen technology are designed to mix lipids and solvent with RNA in the buffer rapidly and consistently to control the self-assembly of RNA LNPs. With typical RNA LNP materials, Spark cartridges are designed to mix with a 2:1 aqueous to organic ratio, followed by a 1:1 dilution. Now let's look at some data generated with the NanoAssemblr Spark. It is very important to determine suitable lipids for the production of efficient RNA LNPs. Here I am showing you an example of screening different ionizable or cationic lipids in low-volume RNA LNP formulations, comparing particle size and encapsulation efficiency. Permanently cationic lipids, here DOTAP and DOTMA, was compared to our proprietary ionizable lipid, denoted here as PNI-ILA. The N-to-P ratio, concentration, and formulation parameters were held constant. The ionizable lipid, PNI-ILA, produced smaller particles and showed lower polydispersity index, PDI, while all of them showed greater than 90% encapsulation efficiency. The next dataset here is a systematic screening of the N-to-P ratio with mRNA LNP formulations. The N-to-P ratio is a key factor in performance, and we tend to recommend the screen early in the drug development process. To determine the effective N-to-P ratio in mRNA LNPs, here we show 3 different ratios. With the lower the number, the higher the RNA in the aqueous solution. As expected, the N-to-P ratio of 4 shows the largest particle size and slightly lower encapsulation efficiency when compared to ratios of 6 and 8. The N-to-P ratios of 6 and 8 behave rather similarly, suggesting an asymptotic limit has been reached, though I would test the biological performance of all of these particles as they are all good quality, and this may impact biological outcomes. LNPs can be used as a platform for delivery of different nucleic acids. Here, 3 mRNAs have been encapsulated to explore the effect of different mRNA lengths. These were GFP around 1 kilobases, Fluke around 2 kb, and Cas9 mRNA around 4.5 kb. The physicochemical properties of all of these RNAs are rather similar at 100 nanometers, exhibiting low PDI and high encapsulation efficiency, indicating that mRNA does not impact key physical attributes of LNPs when manufacturing on the next-gen microfluidic platform. Finally, let's look at a case study using our lipid nanoparticle mix, the Genvoy ILM T-cell kit for mRNA. This lipid nanoparticle kit was designed for the acceleration of T-cell therapies. Genvoy ILM T-cell kit for mRNA is optimized for complex gene editing and gene delivery applications in human primary T-cells. It is available as an off-the-shelf LNP reagent kit, along with a complete protocol of cell culture and LNP treatment. Here is a proof of concept demonstration as the ex vivo manipulation of human primary T cells. These exemplify how we can use LNPs to accomplish complex T cell engineering that enable future therapies. We can deliver a gene, do single or double gene knockouts, and finally do both, edit, then deliver genes, all using LNPs and the NanoAssemblr Spark. Due to the time constraints, I will have to limit only to gene delivery. That said, we wrote an awesome application note that has all of this and much more in extensive detail. This dataset shows CAR mRNA delivery, so chimeric antigen receptor mRNA delivery, either using electroporation or lipid nanoparticles to human primary T cells. CAR expression was measured using flow cytometry 48 hours after transfection. LNPs show a significant increase in percent transfection efficiency when compared to electroporation. With LNPs, we are looking at 85 to 90% expression, while we achieved about 55% with electroporation. The flow histograms show a much more homogeneous expression profile with the lipid nanoparticles, without a significant negative peak that we can observe with electroporation. LNPs also allow for significantly higher cell viabilities. As we can see in the mock condition of electroporation, the method itself impacts cell viability, with both transfection efficiency and viability being an important consideration for the overall yield of a cell therapy product. If you're interested in learning more, please do check out our application note on our website. So how does the Spark enable you to explore novel nanomedicine formulations? The NanoAssemblr Spark is the only instrument that combines the exquisite control of non-turbulent next-gen microfluidic mixing with a microliter process volume in order to enable researchers to reproducibly formulate microgram quantities of nucleic acids into LNPs. The Spark was specifically designed for in vitro ex vivo screening with simple operations and gamma-irradiated cartridges seamlessly integrating into sterile cell culture workflows. The Spark enables on-demand formulations to accelerate the discovery and early preclinical development of RNA lipid nanoparticles. So how can you utilize our technology to accelerate your research? You can encapsulate your RNA in one of our Genvoy ILM lipid mixes and the Spark instrument to achieve a homogeneous lipid nanoparticle formulation. This will significantly improve your workflow, reduce your hands-on time, and increase your overall efficiency. I do want to thank you for taking the time and learning about the NanoAssemblr technology today. For more information on our products and technology, please reach out to us or visit our website. We would be glad to work with you. Thanks again.